Hello friends, welcome to yet another intriguing edition of Biology Made Easy. Today I will bring you a very ecstatic topic, that is the part 2 of the elongation of the DNA replication in prokaryotes. In my previous videos, I have already talked at length about the initiation part and the first part of elongation, where I described extensively about the 10 different subunits comprising DNA polymerase 3 made up of 900 kilodalton of mass, and how does it differ in properties and in processivity from the DNA polymerase 1, and how are different DNA polymerases from 1 to 5 they do pan out various roles. How do they pan out various roles in the E. coli or Escherichia coli? Now we will be talking about the very molecular and biochemical details. How do the nucleotides or the DNTPs, deoxyribonucleotides, get added up, and how does the leading strand and the lagging strand phenomenon stack up? Now let's get down to the business. Now, very first thing, in your books you might have seen a different diagram to this. You might not seen this curved, might not have been able to see this curved diagram. You would have seen a lagging strand diagram and the leading strand diagram, which does not coincide with the trombone loop model. Well, let me explain to you. This is the trombone loop model, and this is the most accepted model in which favors the DNA replication. Why and how? First thing, if you have watched my previous video, I had shown you that the DNA polymerase 3 made up of 10 different subunits is an asymmetrical dimer, meaning that the polymerase would load on in a horizontal way like this. Means one of the core polymerase would load here, another of the core polymerase entire subunits, the alpha, the epsilon and the theta, would be loaded here. Here would be the beta 2 clamp, here would be the asymmetrical clamp loader connected via the symmetrical tau subunits. Now there is only one asymmetrical dimer of DNA polymerase 3. There are no different DNA polymerases which would catalyze the addition of DNTPs on both these strands. So there is only one specific DNA polymerase 3 asymmetrical dimer. So now it just cannot start or initiate the lagging strand in that direction, and for the leading strand it cannot go in this direction. In your previous books you might have seen that lagging strand goes in this direction, leading strand goes in this direction. So if that is really supposed to happen, then what would happen is the polymerase would break off, because the polymerase would get so much stretched that it would lead to the molecular breaking of the polymerase. Thereby it would lose its asymmetrical dimer activity and it would lose the beta 2 clamp and the clamp loader. Thus thereby it would plummet the processivity of the polymerase. So that's not what we are intending to achieve here. So what we are trying to do is, the scientists came up with this model. What happens is, this model keeps the polymerase going in one direction only, doesn't stress the polymerase too much, the DNA polymerase 3 holoenzyme 10 subunit complex too much that it would dissociate and break apart. Now, for the leading strand, it's pretty simple. The first 10 nucleotides would be synthesized by DNAG or DNA primase, which I have already explained to you at length in my initiation part of DNA replication video. Then DNA polymerase 3 would take over, and since this is the 3 to 5 prime strand, template strand, parent strand, 5 to 3 prime, synthesis would be very easy. That is known as the leading strand. It's also continuous without any breakage. It's also known as the continuous strand. Now let's come to the strand which is 5 to 3 prime in direction. Now there is no plausible polymerase discovered yet which can synthesize any strand from the 3 to 5 direction. So there is no polymerase which has the 3 to 5 prime polymerase activity. All the polymerases do possess the 5 to 3 prime polymerase activity. Now what is to be done? How is to be rectified, and how did the nature come up with single or unidirectional follow through or unidirectional movement of DNA polymerase 3 holoenzyme? The answer to that question is, since DNAB is the DNA helicase and DNA primase complex are attached like this in a hexameric manner. Now what happens is, this part, the lagging strand template, what happens is it gets unwound much more than the leading strand template. So this gets unwound much more. Like, it has to be unwound much more, not in the sense that it gets half unwound, but the synthesis of the leading strand is always more or is always faster than compared to the lagging strand. So this is being unwound, you are getting the synthesis done, but this has to be unwound more before the synthesis begins. That is the tricky part here, because when it gets unwound more, what happens is it forms a loop. The DNA loops around, known as the trombone loop model. Trombone is a musical instrument, so known as the trombone loop model. So when it loops around here, the 5' end is here. You can correlate it with the 5' end here. If you look closely, if you do look closely, these two sides, the 5' end of the leading strand template and the 5' end of the lagging strand template, they are in the same direction right now. Just look closely and you can say precisely. Now, then it can serve as the template. Now you can say DNA polymerase holoenzyme does not have to go in that direction. It can start from 5 to 3' synthesis in this direction. The first Okazaki fragment is this one, then followed by this and various Okazaki fragments. Thereby, it negates or it alleviates the need for the DNA polymerase 3 to go in two separate directions or two opposite directions. It would be vectorially impossible. So it has to move in a unidirectional way, DNA polymerase 3 holoenzyme, in order to retain its processivity, fidelity and association with the clamp and the clamp loader. Now what happens is the DNA B primase complex is attached to the tau subunit of the clamp loader. Thereby it does two things. Number one, it increases the speed of the DNA polymerase and causes the switch from DNA G to DNA Pol 3. Next thing, this association also causes the increase in the processivity and prevents the DNA polymerase 3 holoenzyme from falling off, which I had already explained to you in my previous video. What is processivity and how does it negate the falling off of the DNA polymerase 3 from this specific template strands. Now, DNA primase and beta 2 clamp, they do not possess processivity. What does it mean? That every time in the lagging strand synthesis, you can see the synthesis has to be discontinuous. It has to be again reinitiated and rebroken. So every time the primase needs to attach again. So it keeps on falling from the template strand, and every time what happens is during the detachment of the DNA polymerase 3 holoenzyme from here, what happens is the core subunit stays attached to the template, but the beta 2 subunit gets dissociated via the gamma subunit of the clamp loader. It causes the hydrolysis of ATP, thus causing the beta 2 subunit to alter its conformation, to have change in its conformational state, and therefore beta 2 subunit or beta 2 clamp or PCNA in eukaryotes would get dissociated. A new beta 2 clamp subunit always comes for the next cycle of, you can say, Okazaki fragment or discontinuous fragment synthesis. So that's the molecular detail. Now you can see there would be various RNA primers here, because the initiation cannot be done by the 3 to 5' polymerase activity, or there is, you can say, there is no polymerase which can do the de novo polymerase activity of from the 5 to 3' end. So here also we needed DNA G. So here every time an Okazaki fragment is made, since DNA polymerases are unable to catalyze de novo polymerase activity, or it cannot synthesize the polymerization from 5 to 3' direction on its own, it needs a pre from 3 OH group. Therefore you would need the DNA G or RNA primers every time. So you would have multiple RNA primers. But do we need RNA in DNA? No. So one RNA primer from here, multiple RNA primers from the discontinuous Okazaki fragments have to be truncated or have to be removed. How does it happen? We have the 5 to 3' exonuclease activity of DNA polymerase 1 present in only prokaryotes. In my previous video, I had already acquainted you with the fact that there is no DNA polymerase in the eukaryotic system which contains the 5 to 3' exonuclease activity, and they do use the flap endonuclease or the FEN1 enzyme for the removal of the multiple RNA primers. But here the DNA polymerase 1's 5 to 3' exonuclease activity would trim the RNA part from the 5' direction and simultaneously synthesize this 10 nucleotide DNA in the form of DNA by its own DNA polymerase 1's 5 to 3 prime polymerase activity. It doesn't need DNA pol 3 to be loaded again because it's just 10 nucleotides and the processivity wouldn't matter that much, only for 10 nucleotides. Then the last two nucleotides would not be attached because since there would be many primers, the last two would not be attached from 5 to 3 prime. So the attachment of last two nucleotides, just the phosphodiester bond remains, has to be performed by DNA ligase in both prokaryotes and eukaryotes. The only difference lies in the fact that DNA ligase in prokaryotes uses NAD plus as the cofactor, whereas DNA ligase in the eukaryotes uses ATP as the cofactor. Now coming down to the equation of DNA replication. dNTP would be associated with dNMP, which is the to the power n, which is the entire template strand, and dNMP n now is the whatever nucleotides have been added in the daughter strand. So dNMP1 or dNMP to the power n means whatever nucleotides present in the daughter strand, and dNTP means the incoming dNTPs would react in the presence of DNA polymerase, maybe 1, maybe mostly is 3. Magnesium is the important cofactor, and dNMP n plus 1 means the nucleotide or the deoxyribonucleotide would be 1 more, or 1 would be added here, so it would increase the length by 1 nucleotide, and with the concomitant release of PPI or pyrophosphate. So this is a SN2 reaction, substitution nucleophilic 2 reaction, where the 3-OH group present in the daughter strand attacks nucleophilically on the 5-phosphate group of the incoming dNTP. Here I would like to acquaint you with two major things. This dNTP serves two purposes. Number one, it's the substrate. Number two, it is the source of energy from where the reaction takes place and from where the association of dNMP happens with the dNMP to the power n or to the daughter strand, and thereby PPI gets released concomitantly. So that's the equation. It serves dual purpose, very important. And the trombone loop model, it's the latest model as per the research, because the DNA pol 3 has to move unidirectionally in one direction, otherwise it would get detached, it would lose its processivity, fidelity, and it would also lose the beta 2 clamp and the clamp loader. Thereby you cannot have the processivity, thereby you would lose the number, or you can see the rate of the DNA strand synthesis, right? And the number of nucleotides also, it would synthesize less, it would keep on falling off. So that all problems we would have, in order to circumvent those problems, the nature has come up with the trombone loop model, right? So that's about it, about the conceptual prowess required to comprehend this molecular detail, this complex process of DNA replication in prokaryotes. If you have understood this video, if you are able to comprehend, then kindly hit the like button and the subscribe button also. If you have any kind of queries remaining, then do not hesitate to post it on the comment section below, and do subscribe my channel. Thanks a lot. See you soon.